So this is a really quick follow-up on some cool properties of amino acids. So there are three different, so we talked briefly about how amino acids are incorporated into proteins at the ribosome, and that we have 20 canonical amino acids. And so these are the amino acids that if you were to take all the proteins in our body, chop them up, and categorize the amino acids, these are the 20 that we would see most frequently. However, there are some other special types of amino acids that we'll encounter, and there are three different classes. The first class are the amino acids that aren't part of the 20 canonical amino acids, but in special situations, they're incorporated at the ribosome just like one of the 20. And so there are two types of amino acids that are fairly commonly found. One is selenocysteine. And selenocysteine, you'll notice it looks a lot like cysteine, except it has a selenium instead of a sulfur. And so one thing that's cool about selenocysteine is that it has a pKa lower than just plain old cysteine, and this makes it more reactive. All right, so if we have proteins that we want to be more reactive in certain situations, then the selenocysteine might be incorporated instead of a cysteine. So example, if we're in a protein that's dealing with oxidative stress, you might see selenocysteines there. Another example is pyrrolysine. And so pyrrolysine is also in certain bacteria is incorporated at the ribosome. It's just not normally found in humans, and these are considered the 21st and 22nd amino acids. But they're only incorporated under very special circumstances. The second type of amino acids, and Elena was actually talking about this a little bit and why she was talking about lysine was her favorite amino acid, and these are amino acids that are modified after translation. And this is usually for a purpose of some kind of like regulation of the protein. So this amino acid, if you look, you should recognize that it's lysine, and this is actually trimethyllysine. And lysines are actually very commonly found in histones, which are the proteins that DNA are wound around. And if you were to protonate and deprotonate that lysine and mess around with the charge, that could determine how tightly the DNA is wound around the histone and then whether or not it can be expressed. So these are modifications that are put onto amino acids after they're translated at the ribosome, and it's usually for purposes of regulating or turning a protein on and off for a particular purpose. That's the second type. And then the third type are non-canonical amino acids. And so this is trifluoromethyl phenylalanine, or TFMF. And these are amino acids where you engineer the tRNA synthetase and tRNA that code for the stop codon AUG to accept an amino or AUG or not AUG, TAG. So normally that's a stop codon. And so what happens is you engineer the translational machinery of an organism for when they see tag, they put in the non-canonical amino acid. And so that's a way that you can put in kind of a funky amino acid that does a cool job into the stop codon instead of just cutting off the translation of the protein. Another cool thing about amino acids is that certain amino acids have the ability to absorb UV light. So all amino acids are going to absorb in the low 200s because of the peptide bond. So all amino acids are going to absorb there. However, there are only 3 amino acids that have a lambda max at 280 nanometers. And these are the aromatic amino acids, so specifically tryptophan, but to a lesser degree tyrosine, and to an even lesser degree phenylalanine. And so we can actually use these amino acids to measure the concentration of a protein. So if we measure the absorbance of a protein at 280 nanometers, and we could take the extinction coefficient, but we can add up the number of tryptophans to give us the extinction coefficient. We have the path length and the concentration. If we measure the absorbance and we know the extinction coefficient, if we know the sequence of the protein, we can solve for the concentration. And we'll see how this is exploited when we look at protein sequencing, actually. And if you were to look at this chart, take a minute. I kind of already said it, but tryptophan is definitely the one that absorbs the strongly because it has that nice double ring aromatic system. Now, I'd like to take a minute to show you an example of what happens when the amount of amino acids in our bodies get off balance, because it's really important for making proteins, but it can have larger ramifications if we're not getting the amino acids we need. So there are two classes of amino acids. We have essential amino acids, and these are the amino acids that we eat. And we have non-essential amino acids, which are the amino acids that we make. And so in our body, it turns out that phenylalanine, or Phe, is one of the essential amino acids. So we have to eat phenylalanine. We can't make it ourselves. But then our body takes the enzyme phenylalanine hydroxylase and adds an OH onto the ring to make tyrosine. So tyrosine is a non-essential because we can make it from phenylalanine. And so there's a disease called phenylketonuria. And what this means is that the enzyme that adds on the hydroxyl group is knocked out. It's insufficient, so you don't have functional phenylalanine hydroxylase. And so what happens then is, yes, it makes sense that the concentration of the tyrosine will decrease in our body, but the real concern is that you get a backlog of phenylalanine in the body. And so what happens then is people who have phenylketonuria, and again, this is diagnosed in babies. So it's one of the first tests that babies get. They get a heel stick. And what happens is you have to essentially eat a low phenylalanine diet. So if you ever look at foods and it says phenylketonuriacs contains phenylalanine, people who have phenylketonuria need to avoid eating foods that are rich in phenylalanine. All right, now, just a really quick naming convention for proteins and peptides. Peptide is a name we use for a protein or a polypeptide that has less than 50 amino acids. Once we get greater than 50 amino acids, we're considered a protein or a polypeptide. And if we have a short peptide, we can do a naming convention where if we have 2 amino acids, it's a dipeptide. 3 amino acids, a tripeptide, etc., until you run out of prefixes. Once you get past 50, we call it a polypeptide or a protein. And this is just another reminder that the convention for writing a peptide sequence or protein sequence is going from N to C. Now, most proteins, if you have a protein of N residues, you have 20 to the N possible sequences. So randomly speaking, there's a whole different range of sequences that can be occupied. Most proteins typically are anywhere from 100 to 1,000 amino acids. And if you want to estimate the molecular weight of a protein, you can estimate it by multiplying the number of amino acids times 110, because 110 is the average molecular weight of an amino acid. And again, sequence really matters, as we'll see, but you can also see a diversity of other things that can complex with proteins that will make them interesting. And then finally, some examples of biologically active peptides that we see in nature. One key example is aspartame or NutraSweet. And so that's one of the reasons why NutraSweet or aspartame works as a sweetener, because it's actually a peptide, not a sugar. It doesn't have a sugar ring. So take a minute. You'll notice that the amino acids contained there are modified aspartate and phenylalanine. But there are also some other natural examples of biologically active peptides. One is oxytocin, and that actually has 9 amino acids. That's a hormone that's released when a mother bonds with a baby. Another one is bradykinin, and it also has 9 amino acids, and that's an inhibitor of inflammation.